first slide there are my disclosures. And what I intend to talk about tonight, just briefly, is, of course, how we test for these NTRK fusions. And I'll talk a little bit generally about we test for any single genomic driver, and then concentrate specifically on NTRK fusions, and then finish with the ESMO guidelines, which have been recently published on NTRK testing. Next slide, please, Emily. Now, just a little bit of repetition. There's never any harm in that. I know that Carlos already covered these, but these three genes, the neurotrophic tropomyosin receptor kinase genes, they are normally involved with neuronal development and kinetics. And there are three of them, 1, 2, 3 on chromosomes 1, 9, 15, encoding 3 proteins, track A, B, C. And whenever you get 3 genes like that on different chromosomes, it means they're pretty important. And it's quite unusual in this sort of context. The problem from our point of view tonight is that very occasionally these will rearrange themselves and be confused with a new partner to become single genomic drivers, SGDs of neoplasia, and they share that capability with lots of other genes like EGFR, ALK, ROS1, so on and so forth, many of which, of course, we test for in our non-small cell lung cancers. And what happens when these NTRK genes move is that the 3' end becomes fused with the 5' end of their partner, which switches them on. And as Carlos already said, it's quite unusual here because we have a group of very rare tumors in which they are very common drivers. And most of these are NTRK3 fusions with ETV6 as their partner. And here we've got things like infantile fibrosarcoma, mesoplastic nephroma, Spitz tumors, and the secretory carcinomas of the breast and the salivary glands. And in those tumors, they're almost always driven by NTRK fusion. And then we've got a group of common tumors, Carlos already mentioned these, in which they are rare drivers. And that determines, of course, how we test them and what tools we use. Next slide, please, Emily. And generally, if we're detecting single genomic drivers, what we can apply depends on precisely what we're looking for. If we're looking for mutations, insertions, deletions, as we do with EGFR, with KRAS, and so on and so forth, we have a choice. We can either use what's often referred to as single gene or small panel testing, as shown on the left there. And this has advantages. It's quick, it's cheap if you're looking for only 1 or 2 genes. It's very flexible, but it's very limited in its scope. You have to know exactly what you're looking for, and you have very little flexibility, and you can't use these on a grand scale. For that, really, you need NGS-based multiplex testing, and the picture on the right there shows Illumina sequencing platforms, and others, of course, are available. And these are a whole magnitude of complexity greater than the single gene testing platforms. And they have the advantage, of course, of being able to look for numerous genetic abnormalities at the same time with great efficiency, flexibility, and indeed accuracy. But they're obviously more exacting technically. Now, people get very passionate and get behind these different platforms and become great advocates of them. My view is that they're both excellent. I think it's a question of using the right tool for the job. And people get rather confused about the pros and cons, so I'll take a minute to try and explain that with an analogy. If I could have the next slide, Emily. There on the left, you'll see a very familiar Nespresso coffee machine, and many of us have these at home, in the office, or wherever. And these will give you a decent cup of coffee. They're good, they're flexible, they're quick. But you can't use them if you're catering for 10, 12 people, and you're very limited to what you've got in your little capsule. So they lack flexibility, and if all you've got is ristrettos, then you're only going to have that particular type of coffee until you go out and buy some more. So they have advantages and they're okay, but they have limitations. The device on the right, in all its magnificence, that's a Lachimbali professional barista's machine. And whereas the Nespresso domestic device is really the equivalent for single gene testing, the Lachimbali is NGS sequencing. And here, of course, you've got something which has infinite flexibility. It can make enormous amounts of very good coffee, more or less the same time, and you can manage to provide it to many people in a very short time. And this is the kind of thing, of course, you see in Starbucks. So they both have their application. You wouldn't want one of those in your kitchen if all you needed was a quick coffee at 7 o'clock in the morning. So either of those, both of those, has its place as appropriate. Next slide, please. So if we are detecting our single genomic drivers and we're looking for rearrangement and fusions, again, we can also use either so-called single gene small panel testing or multiplex testing. But with fusions, of course, and NTRK typifies this nicely. In that instance, it's very similar to ALK and ROS1. The fusion can be, the rearrangement can be visualized by FISH, fluorescent in situ hybridization. You can see that picture there in the middle of those blue nuclei, because you can label either side of the breakpoint where the chromosome fractures and see the displacement of the gene. And of course, the actual fusion process results in the generation of a protein, and you can detect that by immunochemistry. And there's an example of FISH and subsequent immunochemistry for ALK gene fusion, break apart on the FISH, and strong expression of the fusion protein on immunochemistry. And for those tests, of course, you need a microscope, good solid pathology. Next slide, please. So if we're looking specifically for NTRK fusions, what can we do? Well, it's interesting that the modular systems like the Adilla system are now beginning to move in what they call into what they call panel testing. And they're developing cartridges, as the one shown here, in which you can detect a group of abnormalities at the same time. And their gene fusion cartridge, which is currently still under development and being trialed, is actually capable of looking for ALK, ROS1, NET, NTRK, and RET. So that's rather expanding the capability of our equivalent Nespresso machine, which is interesting. However, multiplex testing is very much the tool of choice here because of its sensitivity. And because if you're using a modular system to look for gene fusions, you've really got to know what your partner is. And if you only have one or two possible partners, or one dominates, like with ALK rearrangement, then it's not a major problem. But when you have lots of partners, as you do with NTRK, then it can become very tricky. So in this particular instance, without question, NGS sequencing is the method of choice over single gene or small panel testing. So you would certainly go there for your big barista's coffee machine. However, we can also, because this is a fusion, because it generates a protein, use FISH and or immunochemistry to test for the fusion. And then on the left is a photograph of separation of signals in the nucleus there at the end of that long blue arrow, which indicates that the NTRK3 gene has become rearranged. And on the right, we can see the protein product of that particular dislocation and fusion as revealed by the Ventana EPR 17341 pan anti-NTRK antibody. Next slide, please. Now, I'd like to say a little bit more about the immunochemistry in particular, because I know we have quite a lot of pathologists out there, and I think this is really very interesting. This particular antibody, which is the one that we've been using, that we favor, this the epitopes it looks for at the C-terminal regions of the three proteins, NTRK A, B, and C. Wild type NTRK protein is expressed, it's expressed all the time, but generally at very low levels. Occasionally, it seems to pop up strongly in neuroendocrine tumors, and that can be a source of confusion. You have to watch out for that. But generally, these antibodies don't pick up wild type NTRK protein because it's at very low levels. Positive controls, as you might imagine, cerebellum, you can also use appendix, the nerves in the appendiceal wall are quite useful, so that's not a problem. And of course, pellets from cells, the cell cultures are also very useful as positive controls. what's particularly interesting is that we've begun to realize that what you see when you do your immunochemistry rather depends on the partner. And we've suspected that this goes on, especially with ROS1 fusions, but never seen it in this sort of way. And I just illustrated that here for you. If your fusion partner is the LMNA fusion partner, and that's the commonest fusion partner with Spitz tumors, then it's usually the nuclear membrane where the protein is expressed. If your fusion partner is TPM3, and this is what we often see when NTRK1 fuses with that particular gene in a range of adenocarcinomas, including non-small cell lung cancer, then the cell membrane is the predominant site of expression. And if your fusion is between an NTRK3 gene and ETV6, which is the fusion that underlies most of those rare tumors I mentioned a few minutes ago, then the expression is conspicuously nuclear. And in practice, most are a mixture of this. That picture on the top there is a poorly differentiated adenocarcinoma of the lung, in which really expression is almost everywhere. It's in the cell membrane, it's in the cytoplasm, it's in the nuclear membrane. And that's actually not uncommon in those sorts of tumors. The one on the bottom there is actually a secretory carcinoma of the breast, in which the expression is strikingly nuclear. And it's interesting, isn't it, that by looking at the immunochemistry, you can predict to some extent exactly what the fusion partner might be. So some interesting biology, I think, around that. Next slide, please. Everybody talks these days about so-called liquid biopsy, a very powerful technique, not yet quite found its place, but gaining traction all the time. My view is that it's still supplementary to tissue. Tissue remains the gold standard. It has advantages, of course. It's clearly less invasive. It presumably is less affected by heterogeneity inherent in a tissue biopsy when you get a small part of one deposit of what might be a disseminated tumor. Although that's not proven, it's intuitive but not proven. And it's very useful for monitoring response and progress because you can draw blood and look for genetic abnormalities that will tell you if a tumor is responding to therapy or indeed relapsing. In terms of NTRK fusion specifically, it's currently very limited as a tool and application is narrow. For example, the leaders in what we might call liquid biopsy, Gardens and Avenio, they at the moment have liquid biopsy assays applicable only to NTRK1 fusions. So we're not really there yet, but I'm sure in time that will be developed and become part of our weaponry in testing for NTRK fusions and monitoring the progress of tumors that are driven by it. Next slide, please. Next slide, Emma, thank you. Okay, so what ultimately do the NTRK, do the ESMO guidelines tell us about NTRK testing? What should you be doing? Now, I tend to avoid putting references in my slides, although I've included this one because this is something which you might like to look at because it boils all this down. Having said that, it's a rather badly written paper and it can be very confusing at first reading. But anyway, here is essentially what they recommend, and there's good sense behind this, I think. If you've got one of the rare tumors in which you strongly expect that NTRK fusion is driving it, the congenital megaloblastic nephroma, fibrosarcoma, the secretory carcinomas, and so on, then you're almost really confirming what you're pretty sure is happening anyway. So FISH is a very good bet here. It's a labor-intensive test, but you can do it quite quickly if you do it only occasionally and it doesn't become a burden. Alternatively, you can use reverse transcriptase PCR, the kind of thing that you have in some modular genomic testing systems, or of course you can use RNA-based NGS. And these are the recommended approaches if you have one of these rare tumors. And indeed in pediatric pathology departments, they've been using FISH for some time in confirming that these rare childhood diseases are associated with NTRK fusion. Now, if we're thinking about common tumors in which NTRK fusion is a rare driver, then we have to take a different approach. And you will see the guidelines will tell you that you have an alternative. If you have access to NGS, which of course we will all do shortly, we do in fact already have access, and we've been testing for NTRK fusions in the Northwest for a while, as I'm sure Helene and Emma will tell you shortly, then that really is the best option. Ideally RNA-based rather than DNA-based. It's a more sensitive application for looking for gene rearrangement and fusion. But you will see the guidelines tell you that confirmation by immunochemistry can and should be used as appropriate. What do they mean by that? They mean that if you have any doubt about the results of your sequencing, then confirming it by immunochemistry to show that there is indeed a fusion protein present is an appropriate thing to do. If you don't have access to NGS, and across the world, of course, NGS is not widely available, it's easy to forget that, then they recommend using immunochemistry to look for the fusion protein. And then if you find expression, find hopefully a way of confirming that by RNA-based NGS. I think what they're really saying is that in an ideal world, you would do both sequencing and immunochemistry. So you know that your evidence of rearrangement and fusion by NGS is genuinely linked to a fusion protein, and conversely, if you have a fusion protein, then you can confirm using NGS that it is being generated by a chromosomal rearrangement and is not spurious and non-specific. So they do make sense when you look at them that way. And I'm not going to say anything more about that in terms of what we're doing here in the Northwest, because Helene and Emma will cover that shortly. Next slide, please. And at that point, I'll leave it. Thank you very much for your attention.